Good evening, everyone. Welcome to lecture 3 for cell cycle and cell division. So in the earlier lectures, we study about what is a cell cycle and what is a mitotic division. Now in this lecture, we are going to study what is meiosis and in which kind of cells usually meiosis takes. So starting with the introduction, the production of offsprings, especially in case of sexually reproducing organisms, it includes a union of two cells. For example, in case of humans also, in case of humans, we know that the fusion of the gamete with the ovum leads to the formation of a zygote. So basically, in case of the organisms where the offsprings are produced by the sexual reproduction, it includes the union of two cells, each with a haploid set of chromosomes. So the gametes, that is the ovum and the sperm, which fuses to produce the zygote, are both the haploids. That is, they both contain the haploid set of chromosomes, resulting in a zygote, which is a diploid. So the doubling of the chromosome number at fertilization is compensated by an equivalent reduction in chromosome number at a stage prior to the formation of gametes. So if we want that this 2n number to be continued in the next generation also, then it is important that before the fertilization or before the fusion, the gametes should have half the number of the actual number of chromosomes which are present in any diploid individual. So if the organism is diploid, the gametes should be haploid so that when the fusion of the gametes takes place, the organism which is formed or the zygote which is formed as a result of fertilization, the zygote should contain the equivalent number of the chromosomes as their parents. So this reduction in the number of chromosomes in case of gametes or this reduction in the number of chromosomes is usually accomplished by the process which is known as meiosis. The term meiosis is coined in 1905 from the Greek word that means the reduction. So it is also known as reductional division because in case of meiosis, the number of chromosomes reduced to the half. So meiosis ensures production of a haploid phase in the life cycle and fertilization ensures the diploid phase. This is how the number of chromosomes are maintained from generation to generation. So basically, before the fertilization of the gametes or before the formation of a diploid organism, the gametes have half the number of chromosomes before getting fertilized. So this process by which these gametes have acquired half the number of chromosomes is known as the meiosis or the reductional division. So without meiosis, what happens if this meiosis does not take place? So if meiosis does not take place, and for example, if the gametes also has the same number of chromosomes as any organism, so what would happen? So if I consider the case of a diploid organism, let's say gamete also has 2n number of chromosomes. So to form or in order to produce or reproduce any organism, the fusion of the gametes should take place. So in that case, what would happen is if the number of chromosomes in the gametes would not reduce to half, then what would be the consequence of that? So basically, it will lead to the formation of a next organism that will actually have 4n number of chromosomes instead of 2n. Therefore, it is really important that before the formation of the gamete or at the time of formation of the gamete, the chromosome number should be reduced to half so that whenever the gamete fusion takes place or like the fusion of the gametes takes place, the number of chromosomes after the fertilization or after the fusion of the gametes comes out to be the same as the number of chromosomes in the parental generation. So this is the importance of meiosis, and this is what what is meiosis and what is the meaning of meiosis. So starting with this, so meiosis, unlike mitosis, is divided into two phases. So it is divided as meiosis 1 and meiosis 2. In meiosis 1 is further divided into substages which are very similar to what we study in mitosis. So meiosis 1 is divided into interphase, then prophase, metaphase, prophase 1, metaphase 1, anaphase 1, and telophase 1. After this telophase 1, then there is again the cell enters into the meiosis 2 stage, and the number of stages in the meiosis 2 includes prophase 2, metaphase 2, anaphase 2, telophase 2, ultimately resulting in a tetrads of the cell, or we can say we have 4 haploid cells at the end of the meiosis. So basically in meiosis 1, as we will study later also, so in meiosis 1, there is a segregation. That is, the homologous pairs are separated, reducing chromosome number by half. And then in meiosis 2, the sister chromatids are separated, producing 4 haploid gametes. So this is how from one cell, these 4 haploid gametes are produced. So basically in meiosis 1, just like how the sister chromatids get separated in during the time of mitosis, in this case, in meiosis 1, instead of sister chromatids, basically what the homologous chromosomes get separated. And after this, the separation of homologous chromosome, then when the cell enters into the meiosis 2 stage, the separation of the sister chromatids takes place like that of mitosis, resulting in a tetrads of cell which are haploid. So starting with one by one with each of the phases. So starting with prophase 1, as with mitosis, the prelude to meiosis includes the DNA replication. So before the prophase 1 also, we have an interphase. So just like the interphase of mitosis where the DNA replication was taking place, but one point to remember there was that the DNA replication was taking place, but the chromosome number was still the same. So in this case also, before the onset of the meiosis, DNA replication takes place. And the pre-meiotic S phase generally takes several times longer than a pre-mitotic S phase. So the timeline for the pre-meiotic S phase is comparatively more or larger than the timeline of the pre-mitotic S phase. Prophase of the first meiotic division, that is prophase 1, is typically lengthened in extraordinary fashion if we compared it to the mitotic phases. So if so basically the stages in the meiosis are comparatively larger, takes longer time as compared to the similar stages in the mitotic phase. Meiotic prophase is then further subdivided into 5 stages. These stages are leptotene, zygotene, pectine, diplotene, and diakinesis. So these are the 5 different stages in which the meiotic prophase 1 is further subdivided. So starting with leptotene, what happens during the leptotene prophase 1 stage? So during the leptotene, chromatin threads condense to form the chromosome. So we know that before at the time of interphase, the chromosome is present like in an unwind manner in the nucleus region. So this chromatin threads condense to form the chromosome, and chromosomes are longest and thinnest during this stage. Chromosomes become gradually visible under the light microscope because unlike chromatin, which are loosely present in the nuclear region, chromosomes are tightly bound structures. So all the chromosomes in nucleus remains directed towards the centrioles. So group of chromosomes in nucleus actually appears like a bouquet, and therefore it is known as a bouquet stage. After this is a zygotene or synaptotene stage. So zygotene stage is also known as synaptotene stage, and it is characterized by pairing of homologous chromosomes or the formation of the synapses. So basically in the zygotene stage, the homologous chromosomes pair. So instead of sister chromatid concept here, we have a concept of homologous chromosome in the meiosis 1. Because in the meiosis 1, there is a separation of homologous chromosome that will actually takes place. And then in meiosis 2, the sister chromatid separation will take place. So in case of zygotene stage, in the zygotene stage, there is a pairing of homologous chromosome, and leading to the formation of a synapses. A structure develops in between the homologous chromosome, which is termed as the synaptonemal complex, and the pair of homologous chromosomes are called bivalents or tetrads. However, these are more clearly visible when we go to the next stage, which is the pectiting. The first two stages of prophase 1 are relatively short as compared to the pectiting stage, which is the next stage. So in the pectiting stage, during this pectiting stage, the 4 chromatids of each bivalent chromosomes. So basically, there are 2 pairs, or we can say there is a pair of homologous chromosome. Therefore, each chromosome further consists of 2 sister chromatids. So we can say that in a bivalent chromosome, where 2 homologous chromosomes are paired with each other, so there is a presence of 4 chromatids. So basically in the pectiting stage, the 4 chromatids of each bivalent chromosomes become distinct and appear clearly as a tetrad structure. So all of these 4 sister chromatids are clearly visible and they appeared as a tetrad structures. One of the very important thing is that in meiosis, there is a chance of variation. Unlike mitosis, where the individuals that are produced as a result of mitosis are similar to their parental generation or to their mother cells, in case of meiosis, there is a process which is known as recombination that helps in generation, that helps in creating the variation. For example, even if we see that we are very much similar to our parents, but not the identical, but we are not identical to our parents. So this is because of various events that happen during the time of gamete formation. One of them is the recombination. So recombination nodules between the non-sister chromatids of homologous pair develop. These non-sister chromatids exchange their parts, that is crossing over. So basically crossing over takes place in this stage, and crossing over leads to recombination of genetic materials on the 2 chromosomes. The genetic material between the 2 chromosomes gets exchanged and resulting into the variations. So crossing over is actually an enzyme mediated process, and the enzyme that is involved in this crossing over is known as the recombinase, since it is leading to the recombination. So recombination between homologous chromosomes is completed by the end of the pectiting stage, leaving the chromosomes linked at the sites of crossing over. So wherever this crossing over happens, the chromosomes are linked with each other through that site. So this is very important, that in which stage the recombination or the crossing over is taking place and which is the enzyme that is involved in the recombination or the crossing over, and why it is important. So crossing over actually, or the recombination, helps in creating the variation between the individuals. So this is very important from the point of view of like genetics, we'll see. So in meiosis, unlike mitosis, where the daughter cells are exactly similar to their mother cells, in case of meiosis, the recombination may happen and there is an exchange of genetic material between the non-sister chromatids of homologous pair and resulting into the crossing over. The next stage is the diplotin stage. The beginning of this stage is recognized by the dissolution of the synaptonemal complex, which is formed in the earlier pectiting stage. The homologous chromosomes start repulsing each other, leading to the formation of a structure which is an X-shaped structure. If we will visualize this structure under the microscope, it will give us an X-like appearance, and therefore this structure is also known as chiasmata. And diplotin may last long, up to the months or years, in oocytes of some vertebrates. So if we look, so this is the very, like in cases of some vertebrates, the oocyte remains in this diplotin stage. The next is a diakinesis. So it is the final last stage of the meiotic prophase 1, and it is marked by the terminalization of the chiasmata. So basically chiasmata start opening into the zip-like manner, and chromosomes are fully condensed, and meiotic spindle is now assembled to prepare the homologous chromosome for separation in the next phase. Centrioles start moving towards the opposite pole so that it can facilitate the separation of the homologous chromosome. By the end of diakinesis, nucleolus disappear, nuclear envelope also breaks down, and diakinesis represents the transition to metaphase. So basically, diakinesis is the stage where the preparation for the separation of the homologous chromosomes takes place. The homologous, there is a terminalization of the chiasmata. Chiasmata opens and the meiotic spindle assembly takes place, and chromosomes start assemble themselves to prepare for the separation in the next stage. Centrioles start moving towards the opposite poles, and the nuclear envelope and the nucleolus starts disappearing. So before moving into the next stage, which is the metaphase 1, this is the summary of the prophase 1. So as I mentioned, that the prophase 1 is divided into the 5 different stages. That is, starting with the leptotene, next stage is zygotene. After this, we have pecitine. After this, we have diplotene, and then final stage, which is the diakinesis, followed by metaphase 1. So as I mentioned, in case of zygotene, the synaptonemal complex start forming. Then there is a tetrad formation. We can see that the sister chromatids are clearly visible and resulting in a structure which is known as a tetrad. Then this leads to after this, there is an egg-shaped structure which is formed in case of diplotene. So this is recognized by the dissolution of the synaptonemal complex which is formed. And after the dissolution, the place where these sister chromatids are attached appears to be an egg-shaped structure which is known as the chiasmata. After this, in diakinesis, we study that in case of diakinesis, this chiasmata starts disappearing, and we can see clearly in this diagram, these centrioles start moving towards the opposite poles. So these, we can see that the centrioles are moving towards the opposite poles, and in the next figure, we have the metaphase 1 stage, where we can clearly see that the centrioles have reached to the opposite poles, plus the mitotic spindles, meiotic spindle fibers are attached to attached in such a way that the homologous chromosomes will move towards the opposite poles. So basically, just like it is very similar to the mitosis. The only difference here to remember is that instead of sister chromatids, in the meiosis 1 stage or in the meiotic 1 stage, there is a separation of the homologous chromosomes instead of the sister chromatids. So the next stage after the prophase is prophase 1 is the metaphase 1. So at metaphase 1, the 2 homologous chromosomes of each bivalent are connected to the spindle fibers from the opposite poles. So these, this is a pair of this is a bivalent which is a pair of 2 homologous chromosomes, and we can see that they are attached to the centrioles at the opposite poles from the opposite side. In contrast, sister chromatids are connected to microtubules from the same spindle pole. So we can see that the sister chromatids are connected to the same spindle poles. Therefore, the orientation of the which is made possible by side-by-side arrangement of their kinetochores. And the orientation of the maternal and parental chromosomes of each bivalent on the metaphase 1 plate is random. The maternal member of a particular bivalent has an equal likelihood of facing either pole. So basically, it is not always like that the maternal chromosome will move towards one pole and the parental chromosome will move towards the other. Unlike, more likely is that both of them can have the equal chance of moving towards any. So there is no biasness in movement of the particular set of chromosome towards a particular pole. So the orientation of maternal and paternal chromosome of each bivalent of metaphase 1 plate is random. The maternal member of the particular bivalent has an equal likelihood of facing the either pole. After this, when the chromosomes are arranged in such a manner that they can be, where the homologous chromosomes are arranged in such a manner that now they can be separated. So just like the anaphase stage of mitosis, we have anaphase 1 stage where the separation of the homologous chromosomes takes place. But this separation requires the dissolution of the chiasmata that holds the bivalents together. So basically, during this stage, there is a dissolution of the chiasmata and the separation of the homologous chromosomes happen. After this, the chiasmata, which is maintained by the protein, which is cohescent between the sister chromatid in regions that flanks the site of recombination. So the chiasmata disappear at the metaphase 1 anaphase 1 transition, and the arms of the chromatids of each bivalent lose cohesion. So with the when they lose this cohesion, which is helping in binding the which is helping in keeping these homologous chromosomes together, due to the due to the fact that these cohesion gets loses, the loss of cohesion between the arms is accomplished by the proteolytic cleavage, that is the cleavage with the help of the enzymes, proteolytic enzymes that cleaves the proteins. So basically these enzymes cleaves the cohesion molecules in those regions, but these enzymes do not cleave the cohesion molecules that helps in binding the sister chromatids. So the cohesion between the joint centromeres of sister chromatids remains strong because this is arranged in such a manner that it is protected from the proteolytic attack. So this is how only the homologous chromosomes get separated, but not the sister chromatids get separated during this stage. So what is preventing them from getting separate is the proteolytic activity that can only act on the cohesion molecules or the cohesin molecules that are present between the pairs of the homologous chromosome, but not in between the sister chromatids because they are protected from the proteolytic attacks. As a result, what will happen is sister chromatids firmly remain attached to one another as they move towards the spindle pole during the anaphase 1. Consequently, when homologous chromosomes separate during anaphase 1, each pole will receive a random assortment of maternal and paternal chromosome depending on the orientation, that which side is the maternal side and which pole how the maternal and paternal chromosomes are arranged at the metaphase stage. So depending on that arrangement, each pole will receive a random assortment. It may either receive it may receive maternal chromosomes or it may receive paternal chromosomes depending on the arrangement in their metaphase stage. So thus anaphase 1 is the cytological event that corresponds to the Mendel's law of independent assortment. So we know that Mendel has given different laws, 3 laws. One of them is the law of independent assortment. So this stage, that is the anaphase 1 stage, is basically the cytological event or is the event that is taking place inside the cell that corresponds to the Mendel's law of independent assortment. And as a result of independent assortment, organisms are capable of generating a nearly unlimited variety of gametes because there is a possibility that in some of the gametes, maternal chromosome moves towards one pole, all the paternal chromosomes move towards the other pole. In other cases, vice versa may happen. In some cases, there might be some chromosomes, some maternal chromosomes that move towards one pole and some paternal chromosomes that move towards that pole. So in that way, there are unlimited variety of gametes that may be formed from a single organism. So this law of independent assortment, if you want to look at the cytological event or the event that is taking place inside the cell, so the stage at which this law is basically helping or this law is actually holding true is the anaphase 1 stage. This is the stage that actually determines that how this law governs and how the different unlimited variety of gametes are formed. So this is the representation of the metaphase 1 and anaphase 1 stage. We can clearly see that this red color chromosomes represents the one side of the like paternal or maternal side, and the blue color represents the other side of the chromosome. So let's say if red color represents the maternal side of the chromosome and blue color represents the paternal side of the chromosome. So we have two sister chromatids for each. And at metaphase 1 stage, both of these are joined, but at anaphase 1 stage, these paternal and maternal chromosome get separated from each other, moving towards their respective poles. So after this metaphase and anaphase stage, we have a telophase stage. And telophase 1 of meiosis 1 produces less dramatic changes than telophase of mitosis. Because after this telophase 1 stage, cell again has to enter into the meiosis 2 phase. So therefore, the events that actually happened in the telophase of mitosis that prepares that, like all the activities that the cells start again getting the nuclear envelope, forming the nuclear envelope, or all the activities like the chromosomes get loosened, just like the chromatin that is present in the nuclear region. So all of these activities does not happen in the telophase 1. Although chromosome may often undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus, unlike that of telophase in the mitosis. So if you remember that in case of the mitotic telophase stage, in case of the telophase stage of mitosis, the cell actually start again entering into the interphase nucleus, where it start forming the cell envelope, or where nuclear envelope, or basically it starts start forming the chromatin. So, or it start extending the chromosome in the form of chromatin in the nucleus. So all of these activities do not happen as such in the telophase 1. The nuclear envelope may or may not reform during the telophase 1. So this is the end of the meiosis 1 stage. And all of these steps are mostly similar. The fact is, in case of mitosis, the separation of the sister chromatids takes place. But in case of meiosis 1 specifically, the separation of the homologous chromosome, or the separation of the paternal and maternal chromosomes takes place. Now, the stage between the two meiotic division is called interkinesis, or we can say the stage between the meiosis 1 and meiosis 2 is known as the interkinesis, and generally it is very short. So this meiosis 1, so whatever we have studied in different stages of meiosis 1 is summarized in this figure. So meiosis 1 is divided into 4 different stages: prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. So prophase 1 basically includes where the chromosome condensation and nuclear envelope breaks down. Crossing over happens in the prophase 1. Then in metaphase 1, pair of homologous chromosomes move towards the equator of the cell. After this, in anaphase 1, homologous chromosomes move towards the opposite poles of the cell by getting separated from each other. After this, in telophase 1, basically chromosomes gather at the poles of the cells and the cytoplasm starts dividing. So this is the meiosis 1. And after this, we have different stages in the meiosis 2. So the stages in the meiosis 2 are also kind of similar. So the first stage is the prophase 2. So we know that the gap between the two stages is known as the interkinesis. So interkinesis is then followed by the prophase 2, which is quite simple as compared to the prophase 1. So if the nuclear envelope had reformed into in the previous phase, in the telophase 1, it has to be broken down again. If it has not formed, then it can proceed further with the prophase 2 directly. So basically, it is comparatively easier to this simple prophase than its earlier prophase 1. Then in metaphase 2, the chromosomes become recompacted and line up at the metaphase plate. So the chromosomes basically get arranged at the metaphase plate, just like the one that happens in the during the mitosis. So, in as we know that in case of metaphase 2, the in case of metaphase 1, the homologous chromosomes get separated, but the kinetochores of the sister chromatids get separated or face the opposite poles in the metaphase 2. And these kinetochores get attached to the spindle fibers with the help of the spindle fibers towards the opposite poles. And now in the next stage, which is the anaphase 2, there is a synchronous splitting of the centromeres, which held the sister chromatids together, that allows them to move towards the opposite poles of the cell. So basically, in prophase 2, in prophase 2, which is much simpler, only the nuclear envelope breaks down if it has formed during the telophase 1 stage. After this, the chromosomes become recompacted and line up at the metaphase plate. And unlike metaphase 1, the kinetochores of the sister chromatids of metaphase 2 now faces the opposite poles, and they become attached to the opposite sets of the chromosomal spindle fibers. Then in the next stage, which is the anaphase 2 stage, this begins with the splitting of the centromeres, thus resulting in the separation of the sister chromatids, and these sister chromatids move towards the opposite poles of the cells. And after this stage is followed by the telophase 2. So in telophase 2, meiosis, basically telophase 2 marks the end of the cell meiotic division, and this is the last stage where that marks the end of the meiosis 2. And in this stage, the chromosomes are once again enclosed by a nuclear envelope. Finally, the nuclear envelope is formed around the chromosomes, and the product of the meiosis are haploid cells with 1C amount of the nuclear DNA. So if the nuclear DNA is 2C, then it will be a haploid cell containing the 1C amount of the DNA. So this is about the different stages of the meiosis 2. And this diagram represents the metaphase 2 and the anaphase 2 stages of the meiosis 2 cycle. So unlike the metaphase 1 and anaphase 1, where we see that the separation of the homologous chromosomes was taking place, the parental and maternal chromosome were getting separated. In this stage, we can see that in case of metaphase 2, the separation of the sister chromatids of the homologous pair is getting separated. So in the paternal chromosome or in the maternal chromosome, whatever the sister chromatids, like the pair of the sister chromatids which was present at the metaphase, which remain compact or which remain attached during the metaphase stage, now gets arranged at the now gets arranged in a plane where these sister chromatids are now ready to be separated from each other. And finally, in the anaphase 2 stage, we see that the kinetochores that has bound to the spindle fibers of the opposite poles are now get separated, and these sister chromatids get separated from each other during the anaphase 2 stage. So with this, after this anaphase 2, which is followed by telophase 2, that allows the formation of the nuclear envelope once again, marks the end of the meiosis 2 stage. So the different stages of the meiosis 2 now involves prophase 2, where a new spindle is formed around the chromosomes. And at the metaphase 2, the metaphase 2 chromosomes line up at the equator in a metaphase plate. After this, the centromeres divide and chromatids move towards the opposite poles of the cells. Therefore, the separation of the sister chromatids happened at the anaphase 2 stage. And lastly, at telophase 2 stage, which is followed by cytokinesis, a nuclear envelope is formed around each set of the chromosome. And in cytokinesis, which is similar to the one that happens in the mitosis, so similar to that, the cytokinesis happens, the cytoplasm divides, and the cell finally divides into the two cells. So this is the representation of different stages of meiosis. The left hand side represents the prophase interface followed by prophase 1, where there is a formation of chiasmata, recombination happens, and there is basically this is followed by metaphase, where the homologous pair of chromosomes are arranged at the equator, which is followed by anaphase 1, which leads to the separation of the homologous chromosomes from each other. That leads to the separation of the pair of homologous chromosomes from each other. And after this, there is a stage telophase, where the nuclear envelope may or may not form, and this telophase is then followed by prophase 2. At the prophase 2 stage, the nuclear envelope, which might have formed during the telophase 1 stage, again gets ruptured and it gets reformed. It gets deformed. And at metaphase 2 stage, the pair of sister chromatids gets arranged at the metaphase plate, and similar to the anaphase 1 stage where the homologous chromosomes were getting separated, now in the anaphase 2 stage, these sister chromatids will get separate from each other. And after this, the separation of these sister chromatids from each other, this anaphase 2 stage is followed by telophase 2 stage, where the nuclear envelope starts reforming, and ultimately these two pairs of cells that are formed as a result of meiosis 1 and meiosis 2 results in the four haploid cells. So from a single cell at the end of the meiotic division, we have a tetrad, which is a pair of tetrad. That means we have four haploid cells, each containing half the number of chromosomes. So this is how the meiotic division takes place. These are the different stages of meiosis. And as we got to know about the significance of meiosis in the first slide itself. So again, meiosis is the mechanism by which the conservation of specific chromosome number takes place. If this meiosis does not happen, if this reductional division does not happen, the number of chromosomes would have got duplicate in the every next generation. So in order to maintain or in order to conserve the specific number of chromosomes in each species across the generations, especially in case of sexually reproducing organisms, where the fusion of the gametes is required to produce the new organism, in that case, meiosis is the mechanism that allows the conservation of specific number of chromosomes. And even though the process results in the reduction of chromosome number by half. So during the process, although the chromosome number reduces by half, but ultimately when these gametes get fused with each other, this result in the formation of the zygote, which actually have the same number of chromosomes as that of the parental generation of that species. Also, we get to know that in case of meiosis, the chances of genetic variability increases due to the process of recombination or the crossing over that takes place, especially in the pachytene stage of the prophase 1. So basically, it also increases the genetic variability in the population of organisms from one generation to the next. And variations are very important for the evolution because variation allows the survival of the individuals in a drastic situations. So therefore, variations are very important for the process of evolution. And also these variations, when accumulated over a large period of time, might result in a different species. Therefore, they are very important in the process of evolution. So that is the importance of meiosis, because without meiosis, this would not have been possible, unlike in mitosis. So just to summarize what the differences between the mitosis and the meiosis, basically prior to both mitosis and meiosis, diploid G2 cells contain pairs of homologous chromosomes, with each chromosome consisting of two chromatids. Now, during mitosis, the chromatids of each chromosome are split apart and separated into two daughter nuclei in a single division. As a result, cell produced by mitosis contains pair of homologous chromosome and are genetically identical to their parents. However, in case of meiosis, the four chromatids of a pair of replicated homologous chromosomes are distributed among the four daughter nuclei, and this meiosis accomplishes this feat by incorporating two sequential divisions without an intervening round of DNA replication. In the first meiotic division, each chromosome that consists of two chromatids is separated from its homologue. As a result, each daughter cell contains only one member of each pair of homologous chromosomes. Now, for this to occur, homologous chromosomes basically pair during the prophase of the first meiotic division, and that does not happen in the mitosis. So as they pair, homologous chromosomes engage in a process which is known as genetic recombination that produces chromosomes with new combinations of maternal and paternal alleles, thereby causing the variation, resulting in the variation. In the second meiotic division, the two chromatids of each chromosomes are separated from one another, resulting in a tetrad or four cells that have a haploid number of chromosomes. With this, this is the end for the chapter cell cycle and cell division. This was all about the meiosis today. In the last lecture we studied mitosis, and in the first lecture we had the introduction about what is a cell cycle, what are the different stages before actually the cell division takes place, how the cell prepares itself for the cell division, and what are the different events that happen during the process of the cell cycle and cell division. So with this, I would like to end this session for today. Thank you so much.